Come to biology, the scientific study of life. The molecular biology of root nodule formation. How does a legume species recognize a certain strain of rhizobium among the many bacterial strains in the soil? And how does an encounter with that specific rhizobium lead to development of a nodule? These two questions have led researchers to uncover a chemical dialogue between the bacteria and the root. Each partner responds to the chemical signals from the other by expressing certain genes whose products contribute to nodule formation. The plant initiates the communication when its roots secrete molecules called flavonoids, which enter rhizobium cells living near the roots. The signal's specificity arises from variations in flavonoid structure, with a particular legume species secreting a flavonoid that only a certain rhizobium strain detects and absorbs. The plant signal activities a gene regulating protein in the bacterium, which switches on a cluster of bacterial genes called node for nodulation genes. These node genes produce enzymes that catalyze production of species specific molecules called node factors secreted by the bacterial cells. The node factors signal the root to initiate the interfection process that enables the rhizobium to enter the root and to begin forming the root nodule. The plant's responses require activation of genes called early nodulin genes by a signal transduction pathway involving Ca to the 2 to the power positive and second to the power 2 positive and second messengers. By understanding the molecular biology underlying the formation of root nodules, researchers hope to learn how to induce rhizobium uptake and nodule formation in crop plants that do not normally form such nitrogen-fixing symbiotic relationships. Symbiotic nitrogen fixation and agriculture. The agricultural benefits of symbiotic nitrogen fixation underlie uptake, uh, up, um, um, underlie crop rotation. In this practice, a non-legume such as maize is planted one year and the following year alfalfa or some other legume is planted to store the concentration of fixed nitrogen in the soil to ensure that the legume encounters its specific rhizobium. The seeds are soaked in a culture of the bacteria or dusted with bacterial spores before sowing. Instead of being harvested, the legume crop is often plowed under so that it will decompose as green manure, reducing the need of manufactured fertilizers. Many plants families beside legumes include species that benefit from symbiotic nitrogen fixation. For example, alder trees and certain tropical grasses host gram-positive bacteria of the actinomycetes group. Right, rice, a crop of great commercial importance, benefits and directly from symbiotic nitrogen fixation. Rice farmers culture a free-floating aquatic fern called azolla, which has symbiotic cyanobacteria that fix nitrogen and increase in fertility of the rice paddy. The growing rice eventually shades and kills the azolla, and the composition of this organic material adds more nitrogenous compounds to the body, to the paddy. Myco, um, Mycorrhizae and plant nutrition. Mycorrhizae fungus roots are modified roots consisting of mutualistic associations of fungi and roots. The fungus benefits from a steady supply of sugar donated by the host plant. In turn, the fungus increases the surface area for water uptake and selectively absorbs phosphorate and other minerals from the soil and supplies them to the plant. The fungi of micro rhizia or mycorrhizae, as shown this figure. Notice that mycorrhizae, in turn, the fungi or fungus increases the surface area for water uptake and selectively absorbs phosphate and other minerals from the soil and supplies them to the plant. The fungi of mycorrhizae also secrete growth factors that stimulate roots to grow and branch, as well as antibiotics that may help protect the plant from pathogenic bacteria and fungi in the soil. Mycorrhizae are not oddities. They are formed by most plant species. In fact, this plant fungus symbiosis might have been one of the evolutionary adaptations that made it possible for plants to colonize land in the first place. Indeed, fossilized roots from some of the earliest plants include mycorrhizae. When terrestrial ecosystems were young, the soil was probably not very rich in nutrients. The fungi of mycorrhizae, which are more efficient at absorbing minerals than the roots themselves, would have helped nourish the pioneering plants. Even today, the plants that first become established on nutrient-poor soils, such as abandoned farmland or eroded hillsides, are usually mycorrhizae rich. The two main types of mycorrhizae, the modified roots formed from the symbiosis of fungi and plants, take two major forms: ectomycorrhizae and endomycorrhizae. In ectomycorrhizae, the mycelium mass of branching hyphae forms a dense sheath or mantle over the surface of the root, as shown in this figure. Fungal hyphae extend from the mantle into the soil, greatly increasing the surface area for water and mineral absorption. Hyphae also grow into the cortex of the root. These hyphae do not penetrate the root cells, but form a network in the abublist or extracellular space that facilitates nutrient exchange between the fungus and the plant, compared with uninfected roots. Mycorrhizae are generally thicker, shorter, and more branched. They typically do not form root hairs, which would be superfluous given the extensive surface area of the fungal mycelium. About 10% of plant families have species that form ectomycorrhizae, and the vast majority of these species are woody, including members of the pine, cypress, oak, walnut, birch, willow, and eucalyptus families. In contrast,